Hello, welcome to S Chemistry. I am your instructor Dr. Suhas Gauri, and today I will be teaching BSc 5th semester Physical Chemistry course on electrophoresis. Here is the outline. So in this class I am going to cover the types of the electrophoresis techniques, which are there following two: moving boundary electrophoresis and zone electrophoresis. Followed by I will cover the application of electrophoresis. So let us start with the electrophoresis technique. First, let's get introduced about who invented the electrophoresis technique. So the electrophoresis is defined and discovered by Tisselus, the name of scientist Tisselus. So Arjen Tisselus was a Swedish biochemist who won the Nobel Prize for Chemistry in 1948 for his work on electrophoresis and adsorption analysis, especially for his discoveries concerning the complex nature of the serum protein. So he invented this electrophoresis technique, and using this technique, he uh, di- discovered concerning the complex nature of the serum protein. So he isolated that protein, serum protein, using this electrophoresis technique. So let's focus on moving boundary electrophoresis. So what is mean by moving boundary electrophoresis? So in right hand side you can see one figure which is about the moving boundary electrophoresis. That is, this technique is defined and discovered by Tisselus. This method used until 1950 involved a U-shaped tube cell with anode and cathode. So as you can see in this figure, you can see the shape of this tube is a U-shaped tube. This is made up of glass, and two anode electrodes will be there. One is anode and one is cathode. So this anode and cathode generally dip into the this column of U-shaped tube. So the protein solution A, B, C, D, these are the ions of the proteins, where he was put into the bottom into the U-shaped tube, and above that buffer solution was kept above this solution. So that's how the arrangement was. So it has two minus ion and two cation and two anion. So like this is a mixture of protein solution. So on the application of electric potential, so if you apply this electric potential of the two end of the U-shaped tube, so you will get the, so your ions, that is negatively charged ions A minus and C minus, these ions will migrate towards the anode, and B plus and D plus will migrate to the cathode part. So that's how you will able to see over here. So this is a pure A, pure C, this will over here. After applying the electric potential, so this negative will over move towards the anode, and positive ions will move toward the cathode. So that's how will apply. So movement of solution particle under applied electric potential is called the electrophoresis. So here electrophoresis will happen after applying the electric potential. So both ions will get separated out due to applied of electric potential. So one figure is about before and after applying the after electrophoresis. So these are the two basic differences will happen over after reaction. So this concentration at electrode can be measured by a refractometer. So what are the moved ions towards the anode and cathode, that will measured, that concentration will be measured using the refractometer. So at boundary of two layers in U-shaped tube, so two different layer will be there. So here ions and here positive. So cation and anion will be there. So using refractometer you can measure the concentration of the U-shaped tube. So moving boundary electrophoresis is ion with high mobility migrate faster in relation to other ions with low mobility. So, who will mobilate faster? So, in this technique, so higher mobility migrate faster. So those have a higher mobility, they will migrate faster into the compared to the relation to the lower mobility ions. So depending on the ions you can decide which will move faster and low mobility. So this movement of the solution particle under electric potential, this is the electrophoresis or cataphoresis. These are the basic definition, and this figure is about the before electrophoresis and B is the after electrophoresis. So that is the basic difference. So now we will move to the next electrophoresis technique, that is zone electrophoresis. So in zone electrophoresis, first we will see why this technique is important. So as you can see and right hand side, this is about moving boundary electrophoresis. So one drawback was there of this moving boundary electrophoresis. The technique, in fact, it the separated ion are likely to mix with each other due to the presence of convective current present in the U-shaped tube. So here, due to electric potential, these ions was separated out. So after separation, so there will be particular uh, current was moving. So that current we generally talk about. So where current we generally will appear, where temperature difference will be there. So due to this electrophoresis technique, there will be difference in here high temperature and here low temperature. Due to difference in the temperature in solution, you will have particular current, that is convective current. So due to this conventional current, so after separation also you will get the mixture. So due to this current they are always getting mixture. So they come up with the idea where to avoid this problem, mixing problem, uh, the stable supporting media like filter paper, cellulose acetate or silica gel are used to prevent the migration and mixing of the electrophoretically separated ions. So they use one kind of filter paper that we will come to know here. So they just put in between so that the mixing of ions won't happen. So you just remember the name of this paper. This is filter paper of made of cellulose acetate or silica gel. So these are the material of the filter paper they generally use for the prevent the mixing of the electrophoretically separated ions. Now we will see. So this is the zone electrophoresis. You can think of it. This is the figure of zone electrophoresis. So here you will see one glass plate. On top of that you will be having support. For the support you will be having your sample A, B, C will be loaded in the here, and this is the Buffel's filter paper. So both side you will be having cell, electrophoretic cell, where in that in in cell you will be having anode, in another cell you will be having cathode. So these anode and cathode are connected to the negative and positive ions of the electric current, electric potential, and this cell contains some solution will be there, that is made up of buffer solution. So this is the basic diagram of zone electrophoresis. So first working principle of zone electrophoresis is, so the cell contain a buffer solution with a specific conductivity. So first is the we are talking about the buffer, which is contained in the electrophilic cell. So it has a specific conductivity. So this cell contains buffer solution with the specific conductivity. You will be having specific conductivity, and then the sample mixture is placed at the center of the support medium, that is filter paper. So you will be having this buffel filter paper where your sample will be uh, placed over here in the center. So this is the second part. So third part is about the electric field, so that is applied to this setup. So the working of the electric field is to separate out the two different kinds of ions. So that is the working principle of a anode, electric field. So now we will think fourth part is, we will see the fourth part. So fourth part means the different component of the mixture migrate towards the electrodes at varying velocities. So one is positive electrode, another is negative electrode, that is cathode and anode. So after apply of electric application of electric field, you will be able to out the here mixture, your sample contain mixture. So with different velocity it will move towards the electrode. So this is due to different components present in the mixture. So that is cation or anion, it will move according to its low mobility or high mobility ions will be there. So that will be differ by the velocity. So fifth point is the difference in the migration speed is due to the varying mobility of the individual ions. So due to different varying different mobility of ions, so that's why we are always getting for different ions, different individual ions. So sixth point is the molecular are separated in the order of their mobility. So at given in figure 3.3 they have shown the uh, different like order of where C, A and B are coming out into the mixture. So first figure is about the initial mixture of ions. So this is the concentration of all at one place, all molecules are in one place, all ions are in one place. So if you apply the electric field, first you will get uh, first you will get C as a pure compound, then A, then B. So that's how you will get the first separation, because C adds a lower concentration, B, then A and then B. So because C has higher mobility, so that's why it come faster. B has least mobility among ABC, so that's why it came at the end. And A has moderate mobility, so it comes in between to the while during the separation of the uh, zone electrophoresis, during the separation of ions. So these are the molecules are separated in the order of their mobility. So C first it will come C, then A, then B. So that is the order of molecules. Now we will see like what are the parameters which are affecting the zone electrophoresis. So first is influence of substance participation. So whatever you are adding as a sample or substance, that is our interest of sample. So influence of substrate participation is always there. So the shape of zone in electrophoresis is affected by how substance engaged in absorption equilibria. So what are the sample that are going to absorb into the substance, these are the substance electrophilia. So now the second is the importance of separation time. Second point is importance of separation time. So the duration of separation plays a crucial role in the process. So here if we are able to separate out two different mixture of the sample, then time only crucial part is the time. So as long as your separation time is well resolved, so you will get the well separated ions. If your time is very limited, then you will get the mixture of ions like this over here. So separation time is also very crucial, plays crucial role. So third point is analogy to chromatography. So zone electrophoresis, this technique is similar to elution method used in a chromatography. So where in chromatography for separation we use the elution method. So this is similar to the chromatographic technique, elution chromatographic technique. So what is the fourth point? Fourth point is the density gradient stabilization. So what are the support system you are using in this electrophoresis? So conducting electrophoresis in a density gradient column prevents convection current, which help maintain the separation of ions. So here in previous slide we have seen the separation, what are the ions will are separated due to electric field, but due to convection current they are getting able to mix. But instead of that, if you put a sample, or here in conducting electrophoresis, if you put the density gradient over here, then in that column you can prevent this convection current mixture of sample. You can prevent the mixture of sample or mixture of ions by using the density gradient column. So this is the type of column. If you use this material density gradient column, then you can prevent and able to well resolve the separation of ions, or it will help to maintain the separation of ions. The fifth parameter is the density gradient barriers. What are the density gradient barriers? So examples are the sucrose, glycerol, and ethylene glycol. So these are the type of density gradient barrier or material of density gradient barrier. Why this is important? Because it gets solubilized but it not get ionized. So that is the sixth part. So this is the sixth parameter. So the barrier dissolve in water but not do not ionize. So meaning they are not participating in the electrophoresis, since only ionized substance can move electrophoretically. So these sucrose, glycerol, and ethylene molecules, ethylene glycol, this molecule generally used only for the density gradient, to create the density gradient barrier. But this gets solubilized but not ionized. So this is the best quality of this density gradient barriers of this molecule. So they do not participate in electrophoresis technique, so they don't move because they are not charged. So that are they are not ionized substance, so that's why they are insoluble, they are inionizable. So that's why, so it has a use of that. So now we will see the application of electrophoresis technique. So what are the two techniques we have seen, moving boundary electrophoresis and zonal electrophoresis? We will see where this what are the application of this electrophoresis technique. So first application is, so this is used for the separation and analysis of proteins, essentially in biochemistry for research and diagnostic. So this is generally used for the analysis of protein. If you put protein, mixture of protein, then you can easily separate out, because it has different, has each protein has different molecular weight and molecular ions. So using based on that you can separate out. Another application is, this technique is essential for separation of DNA and RNA fragments. Suppose you want to do study on DNA and RNA fragments, so for that, so this is very important in terms of genetic research. Third application is So this is used in analyzing the serum protein. So in serum also contain protein or enzymes, generally we use to diagnose the diseases, any if any animal or following is any diseases. So you can take that sample, protein sample or serum sample, you can isolate that protein or enzyme, and which is responsible for the particular diseases. So this is very good technique to diagnose diseases. Fourth application is the useful in identifying different isoenzymes, adding in metabolic studies. So enzymes, so your protein can be enzymes or isoenzymes, so depending on what are that, so that can be used as a separating tool or identifying different types of isoenzymes. So generally that is useful in metabolic studies. The fifth point in application is that is employed in pharmaceutical industry for quality assurance and biopharmaceutical. Suppose you have synthesized particular enzymes which is responsible for the metabolic study. If you have created any enzyme or isolated, so for that quality purpose also this electrophoresis technique is important. The sixth application is from the point of to analyze the environmental sample. Suppose in environment, in an atmosphere you have different types of pollutant mix. Suppose you want to analyze that toxin or pollutant chemicals. For that also you can use this electrophoresis techniques. So these are the six application of the electrophoresis. So here we come to end of this lecture. So these are the references for the images, and for the content you can follow these books. So I thank you for your kind listening. See you in next lecture. Till then, happy learning.